everyone, this is your educator Priyanka Thakur. I welcome you all on our YouTube channel Gurmantra Shiksha Ka Granth. Today in this video we are going to talk about genetic code. We are also going to discuss a few numericals and very soon you are going to have a practice session, and in that session I'll be giving you some MCQs, some numericals on genetic code. So do check out this video and please keep liking, sharing, and do subscribe the channel. Okay, let's start with the genetic code. Genetic code. Let's talk about general features of genetic code first. Genetic code is a triplet code. Fine. Each genetic code is a triplet code. At least 3 nucleotides are required to specify one amino acid. If I'm saying methionine, methionine is an amino acid and is encoded by AUG. It means 3 nucleotides. Adenosine will be attached to pentose sugar by glycosidic linkage. Similarly, UNG. Fine. So AUG will be encoding methionine, but there are 3 nucleotides. So to specify one amino acid, at least 3 nucleotides are required. So genetic code is unambiguous. Unambiguous means each triplet specifies one amino acid only. Like AUG is going to specify methionine only. It will not be specifying any other amino acid. So such a nature of codon is called unambiguous. Commaless. You will see no internal punctuations in the genetic code. Okay. Degeneracy. It means that one amino acid can be coded by more than one codon. Do not get confused in between unambiguous and degeneracy. Okay, unambiguous means one amino acid. Okay, each triplet, each codon will be encoding one amino acid. It will specify only one amino acid. But degeneracy means one amino acid can have more than one codon. Okay, so the different codon for a given amino acids are said to be synonymous. If a particular amino acid is having more than one codon, then those codons will be called synonymous. See the amino acid and their synonym codons. Remember all these. Okay, you have to learn this table. Why? Because it will help you in doing some numerical problems. That's why you have to learn this table. Fine. Okay, see, leucine, serine, arginine. These 3 will have 6 synonymous codon. Leucine, serine, arginine, and glycine, proline, alanine, valine, threonine. They will have 4 synonymous codon. Isoleucine will have only 3, whereas phenylalanine, tyrosine, cysteine, histidine, glutamine, glutamic acid, asparagine, aspartic acid, lysine will have 2 synonymous codon. And methionine and tryptophan are the amino acid which have only one codon. So we could say that degeneracy is not applied on these 2. Okay, methionine and tryptophan, they will have only one codon, while leucine, serine, arginine will have 6. You have to remember this table in order to solve your numerical problems. Fine. Okay, I'll be giving you the one in this this lecture only, and later we will, in the practice session, I'll give you more and better problems. So next, a genetic code is non-overlapping. It means that successive triplets are read in order. Let's have an example. AAT, GCT. It's a sequence of DNA. The first codon AAT will encode for one amino acid. Okay, AAT is going to code for one amino acid. GCT will be encoding for another amino acid. So it's non-overlapping. Okay, it's non-overlapping. This is what we call non-overlapping of amino acid. Okay, if it is having UGC, GU is like that, then UGC, UUU, then UGC will be encoding for different amino acid, UUU will be encoding for different amino acids. So some amino acids contain start and stop signals. For example, AUG, which is a start codon, encode for the methionine. Okay, it acts as a start codon, and UAA, UAG, UGA, these are the stop signal codon. These three codon does not code for any amino acid. Remember, we are having the 61 amino acid and 3 are the nonsense codon, or we say stop codon. Total 64 codons we are having. How? See, UAA, there are 3 triplet nucleotide. How many nitrogenous bases are there? 4. So, how many possible nucleotide could be present at one position? 4. 1, 2, 3. 4 into 4 into 4. So 4 into 4 into 4 becomes 64. So we assume that there are about 64 genetic codes, out of which 3 are stop signal codon and 1 is start codon. So we ignored these 3 and we call there are 6 coding codon. Actually there are 61 codons which actually encodes for an amino acid, and these 3 are nonsense codons which are not coding for any amino acid. But see, genetic code is said to be universal. It means that same genetic code is used throughout all life forms. Like if AUG is encoding for methionine, it will encode for methionine in all kind of life forms. But there are a few exceptions, like UAG, which is a stop codon. It can code for pyrrolysine, which is our amino acid, and UGA is also a stop codon. It may code for selenocysteine. Let's see some other exceptions. See, UGA can code for selenocysteine. Universal code, it is a stop codon, and unusual codon that we see is it can encode for tryptophan. UGA can code for tryptophan in mycoplasma, spiroplasma, and mitochondria of many species. Okay, UGA can encode for selenocysteine and for tryptophan. It encodes for tryptophan in case of mycoplasma, spiroplasma, and mitochondria of many species. UGA, you can see in euploids, it codes for cysteine. Okay. UAA, UAG, the remaining stop codon, they both can encode for glutamine in acetobularia, tetrahymnia, and paramecium, etc. UAG can also encode for pyrrolysine. You have to learn all these things, okay? CUG, which generally encodes for leucine, will be encoding threonine in yeast mitochondria. So, learn this table. You need to learn this, fine? Okay, this is the coding dictionary. You will see the first position 5 prime and the third position 3 prime, and this is the second position. Okay, you have to learn this thing, yeah, because it will help you in solving many numericals, number of numericals. U, phenylalanine. Okay, 1 U, second position U, third position U, first position U, second position U, third position C. First position U, second position U, third position A. I hope you understand how you should be coding, how you should be checking this dictionary, and how will you be able to make it yourself. Okay, so you can make it yourself if you are having the time, but still you have to remember which code encodes which amino acid. Like UUC will be encoding phenylalanine, UUA, UUG will be encoding leucine. Sometimes we get confused, like if two amino acids, like two bases are same, we forgot to consider the third one. Usually we say like UUC and UUU. The third position is a wobble position, and it is not having that much effect. But in some cases you have to learn, you have to remember the thing that UU nucleotides are not always going to encode for the phenylalanine. They may be encoding for the leucine. Fine. Okay, like leucine is having 6 nucleotides, phenylalanine only 2, isoleucine is having 3, methionine 1, valine 4, serine 4, proline 4, threonine 4, alanine 4. See, serine, proline, threonine, alanine. Serine, proline, threonine, alanine. How many? 4. Okay. Glycine, how many? 4. See, tyrosine only 2, these are stop codon, histidine 2, glutamine 2, asparagine 2, lysine 2, aspartic acid 2, glutamic acid 2, cysteine 2, arginine 6, and serine is actually having, I think, 6, yes, it is having the 6 serine, and glycine 4. You have to remember this. Oh, if you're going to learn this table, do comment in the comment section. I'll be giving the question in the practice session on this table if you are learning this, only in that case. Otherwise, I'm not giving any kind of numerical. Okay, so what is codon bias? See, like you say, a particular teacher is biased for a particular group of students, just like that. Okay, codon bias, it refers to the fact that different organisms have differences in the frequency of occurrence of synonymous codon in their coding DNA, meaning that some codons are frequently used in particular organisms. See, there are many synonymous codons, one amino acid is encoded by 4 codons or 6 codon. All these are called synonymous codons. So in one organism, some of if one amino acid is encoded by 6 codons, then in one organism, frequency of 3 codons will be more than that of the other 3, and in some other organism the frequency of other 3 codons will be more than the other one. Okay, I hope you understand. So this is what we call codon bias. Okay, here you are having a question: if a peptide has the sequence threonine, alanine, glutamine, glycine, proline, histidine, asparagine, indicate why more than one nucleotide sequence is possible. Okay, you have to answer this: why more than one nucleotide sequence is possible. Fine. And second one is calculate the number of possible nucleotide sequences. What you have to answer me: calculate the number of possible nucleotide sequences. If you are able to answer this question, stop the video, answer the question in the comment section. If not, then here is your answer. You can check out your answer. First answer become: due to degeneracy of codon, you will see many nucleotide. Okay, and threonine, alanine, glutamine, glycine, proline, histidine, asparagine. Okay, so for the second question, you have to check out threonine. Will be having 4 synonymous. See, where is threonine? Let's check out the threonine. Where's threonine? I couldn't find it. Oh, I couldn't find it. Oh God. Where is? I think you can see it, but I don't know why can't I. Threonine is having 4. This one is, oh, here it is. Threonine and glutamic, the alanine, how many alanine? 4. Glutamic acid, this it is 2, and glycine will be 4. Okay, proline will be having, I think, 4. Yes, here it is. Histidine will have 2, and asparagine will also have 2. Here it is. Okay, and the totals become 2048. That's how you're going to solve such kind of questions. Fine, I hope you understand it. I hope you got it now, and you are not facing any kind of problem while doing it. If you have, you can tell me. Okay, fine. How the code was cracked, how it became possible to crack the code. See, first of all, it was Nirenberg and Mathai. Okay, Nirenberg and Mathai in 1961, they used a cell-free culture for elucidation of codon. Okay, they wanted to decipher the genetic code. That's why they used, what did they use? Tell me. A cell-free culture. They didn't use any cell. Instead, they used a cell-free culture. They used RNA template, ribosomes, nucleotide, amino acid, stabilizing agents, and energetic. Actually, they used everything that is required for the synthesis of RNA, that is required for the translation of proteins, that is required for the generation of amino acid. Everything they used. Okay, they used homopolymer of RNA sequences like UUUU, CCCCC, AAA, like this. Okay, they didn't use the GGG, but they do use the UUU, AAA, and CCC at that time. Fine. They were able to synthesize RNA due to the enzyme polynucleotide phosphorylase. This question should have come to your mind. How they were able to synthesize a genetic material without a cell in a cell-free culture. They actually used polynucleotide phosphorylase. Okay, this enzyme This enzyme can catalyze the synthesis of RNA polymer without any template strand. And in some particular physiological conditions, this enzyme can also cause degradation of RNA onto nucleoside diphosphate. Okay, see how this polynucleotide phosphorylase can help in the generation of an RNA. It will be using nucleoside diphosphate like adenosine diphosphate, guanosine diphosphate. It will be taking one nucleotide. It will be removing the phosphate group, and then one nucleotide will be there, and then polynucleotide phosphorylase is the enzyme which will be able to catalyze the synthesis of RNA polymer without any template strand, but we do need some nucleoside diphosphates. Fine. Nierenberg and Mattei, they found this enzyme polynucleotide phosphorylase in E. coli cell-free medium system made uridine diphosphates. Okay, they found out that this particular enzyme made uridine diphosphate if this polynucleotide phosphorylase is provided with uridine diphosphate, then it convert it into a poly-U mRNA. It will make a poly-U mRNA which will be encoding phenylalanine residues. Okay, we will get phenylalanine residues from such kind of mRNA. Okay, they also reveal that poly C codes for polyproline. It means C is going to encode for proline. Poly A like AAA will be encoding for lysine. I hope you understand it now. Fine. Okay, that's how the code was first cracked. Nierenberg played a very important role. Okay, so then they tried to synthesize the mixed copolymer. Okay, so they used mixed copolymer for the deciphering of the code. They used two or more different ribonucleoside diphosphate like UDP, CDP, GDP, ADP. They used in combinations like AC, GC like this, okay, to form the artificial meshes. They said that, they were asked, why are you doing this? They said that if we know the relative proportion of each type of ribonucleotide diphosphate, then we will be able to know the frequency of each type of nucleotide occurring in the mRNA. Okay, like see, if you know that how much proportion of UDP like ADP you are going to add and how much you are adding CDP, you will be able to know how many, what will be the frequency of such kind of nucleotides in the mRNA. Okay, let's take an example. If polynucleotide phosphorylase is presented with a mixture 1 part of ADP and 5 part of CDP, then it will make polymer in which total become, okay, 1 by 6, 1 part of ADP, 5 part of CDP, it becomes 6. Okay, 1 by 6, 1 part of ADP, 1 by 6 of residue are adenylate and 5 by 6 are cytidylate. So now we understand that 5 by 6 will be cytidylate, 1 by 6 residue will be adenylate. Then there will be more polymer of CCC. Okay, there will be more nucleotide having CCC. There will be more sequences in which we will see CCC, and then we'll see 2C and A because they are more cytidylate. There will be a few sequences having 2A and C and very few sequences having only AAA. And now we will be able to know the frequency of these sequences. How? Let's see. 3A, which is going to be least, so you can check it out here. What you have to do, you have to check the probability of occurrence of any triplet. You know that it is 1 by 6 and 3. So here you get 0.4. Final percentage will be 0.4. Possible triplet is going to be only AAA. If the ratio is 1C to A, you have to find out the possible composition. Okay, this is the possible composition. Now 2A means 1 by 6 square. 1C means 5 by 6. Here you get 2.3%. And there are 3 possible triplets with 2A and 1C, then 3 into 2.3 becomes 6.9. Similarly, you can know the frequency of other possible nucleotide as well. Okay, you have so you understand now. You have to remember what is the composition of nucleoside diphosphates, and you will be finally getting the frequency of relative frequency of nucleotide in the mRNA. Get it? You will be, I'll be giving you such a numerical in the practice session as well. Fine. Okay. Govind Singh Khurana also did a great job because he was able to synthesize a long RNA molecule. That long RNA molecule consisting of sequences repeated many times. Actually, what did he do? He made di, tri, and tetranucleotides. Then he replicated them, and then he joined all these nucleotides with the help of enzymes, and then he used such long nucleotide to decipher the genetic code. Nirenberg and Khurana were awarded Nobel Prize for this discovery as well, because Nirenberg was the first to start it, and Hargobind Khurana was the person who was able to decipher the genetic code more accurately. Okay, so now what about this Wobble hypothesis? It was devised by Francis Crick to explain the possible cause of degeneration. I will be giving you an example to understand what is Wobble hypothesis. What exactly it says? It says only first bases of codon have a precise pairing with the basis of the anticodon of tRNA. Okay, first codon, the mRNA, only first bases of mRNA codon have a precise pairing with the basis of anticodon of tRNA. Okay, while the pairing between third base may wobble, that is sway or move unsteadily, and this phenomenon permits single tRNA to recognize more than one codon. Therefore, although there are 61 codons for amino acid, the number of tRNA is far less, which is due to wobbling. Number of tRNA is approximately 35 to 45. Let's see it with an example. See, base in anticodon is G, the base in codon will be U or C. See, in anticodon it's what? G, and in codon it will be U or C. It means that AG will be paired correctly to U, C, and G will be paired to C or U. For C there will be G. Okay, if the base in anticodon is C, then base in codon will be G. But if base in anticodon is G, then the base in codon can be U or C. Okay, if A, it will be U, but if U, it will be A or G, and for I, it will be A, U or C. So this position toward 5' end of anticodon is called wobble position. Okay, this will be responsible for the degeneracy. Okay, this is the cause of degeneracy. So I hope you understand each and everything. I hope you understand the Wobble hypothesis. What is the Wobble position? This third nitrogenous base is actually the third base pair is the Wobble position, which will be responsible for the degeneracy. And you know that what is degeneracy. You can relate the Wobble hypothesis with the degeneracy. So do remember all the codons and the tables, so that you will be able to do a number of numericals. That's all for today. You can follow us wherever you want, but just take care of yourself. That's all I wanted to say. Thank you. Bye bye. Take care.